How do I work the clicker? This one? Okay. Okay, awesome, thank you. Okay, so first I'd like to start off with a disclaimer that this is neither new nor is it my research, but I find it fascinating how science and technology continue to revolutionize the field of medicine. Today, I'd like to tell you the tale of 3 scientists and a tiny fluorescent protein. And I actually just Googled Asian baby, and these are the pictures I came up with. Anyway, so once upon a time, there were 3 scientists who were born, and they could have never known the impact that they would make on the world of science and the world in and of itself. And our story begins with Osamu Shimomura, who was born in Kyoto, Japan, and began studying bioluminescence in 1959. And by 1961, he had discovered, or isolated, the green fluorescent protein from the jellyfish Aquaria victoria. And his research wasn't picked up until almost 30 years later, where our story picks up again with Martin Chalfie, who was sitting in a lab meeting and heard about this protein called GFP and thought, this might be a really interesting protein to use to study gene expression in C. elegans. And for those of you who don't know, there's these little tiny transparent worms that scientists sometimes use to study various cell functions. And so, lo and behold, he was able to study gene expression in C. elegans and published a paper on it in 1994. And that's where Roger Y. Tsien and his lab and his colleagues come into the storyline. And Roger Y. Tsien always had an obsession with pretty colors that started way, way, way back when he was 8 years old doing chemistry experiments in his backyard in New Jersey. And he loved making various colored solutions, and apparently the chemistry set that you could buy at any regular store that one of our presenters showed on an earlier slide, it wasn't challenging enough for him, so he was already doomed for greatness. Doomed? And so pretty colors. He and his colleagues thought, now I wonder how we could change the structure of this protein, or if we change the structure of this protein by making small, tiny changes to the amino acids, if it would change the color of the fluorescence that this protein emits. And lo and behold, it did. And over the next decade, he and other collaborators got together and they were able to come up with the entire visible spectrum of the rainbow in fluorescent proteins. Pretty lights. And this became a fascinating discovery in the sense that the next move that they made was to study various diseased tissues and regular tissues using these fluorescent probes. So now this is where my props come in. This is fun. Okay, so you can think of our entire bodies as being composed of different types of tissues. And in those different types of tissues, you have different cells composing those types of tissues. And in those cells, you have different components. And you have a bunch of, not scissors as big as these, but you can think of enzymes as tons of tiny scissors in your body, cleaving proteins, breaking everything down, right? So that's what their intent was to do, is to make fluorescent probes that target specific enzymes in specific cells and then fluoresce at a specific wavelength. So in a specific light, a specific color, blue, pink, green, red, whatever, what have you. So what they did was they designed a peptide. This is a peptide. And this cleavable linker, this yellow part here, is specific for a specific cell. So this enzyme can't cleave this linker when it reaches that cell. But for this peptide, it can. So boom, pretend it's cutting. And all of a sudden, you have the peptide that is cleaved, and it enters the cell, and the cell fluoresces. Voila. And this allowed them to use fluorescence image guided surgery. So whereas before surgeons had to rely on white light reflectance to see various shades of red and pink, now they can see seriously differentiated tissue structures and look at diseased tissue versus non-diseased tissue, and in that way excise the diseased tissue while saving the tissue that's not. So they can cut around nerves, which are highlighted in yellow, and they can cut around muscle tissue and other structures that are not diseased. So this allowed them to study tumors, atherosclerotic plaques, necrotic tissue, so dead tissue. And it's like surgeons over the past hundreds of years have been relying solely on looking for constellations in the daytime sky. And now all of a sudden they're able to pinpoint the stars in the night. Kind of amazing. Anyway, so that leads to my wrap up. This is the future of... Thanks, guys. That leads to the future of minimally invasive surgery. But prevention is the best medicine. Hit it, DJ. Cancer, let me be. And if not, then just let me see. Where it is your boundaries lie. You think to be my God. My hand, my plan is in demand. The light is bright in the night. Let it give you the guidance to advance. Technology for surgeons doesn't have to change. But it is just as they say. Try to stay healthy in the first place. Don't become too complacent. Prevention is the best medicine. Shit. I'm not finished. Antioxidants, fight free radicals, send us anarchists on sabbatical. Don't suffer from protein deficiency. Those amino acids are important to see. Not just for muscle repair, but also for healthy teeth, nails, and hair. Exercise improves circulation. Don't go and get impatient. Stick with it, and you will see how big and fabulous the results will be. On top of improving blood flow in your cells, oxygen will go. Oh, and if you only knew how oxygen makes your cells like new. So